So hi, everyone. So I'm a postdoc in the Anderson Lab here at MIT, and I'll be telling you about AI for LNP design. So just to give a sense of what I'm going to be talking about, first I'm going to be talking about why we might want to do lung gene therapy and what are the challenges involved, and then the approach that I took to it, which is using deep learning to design gene therapy vectors, and then how we can use this to do real-world design of LNPs and do this in really kind of advanced large animal models and show some real promise for the vectors that we designed. So inhaled gene therapy has a potential to treat a really vast array of lung diseases. So that includes monogenic diseases, things like cystic fibrosis or ciliary dyskinesia, as well as more complex diseases like asthma, lung cancer, or COPD. And in all of these diseases where the locus of the disease is in the lung, it would be really great if we could deliver, say, some kind of gene therapy directly to the lung. It has a lot of advantages. You get local delivery to the site of disease, so you're getting a better therapeutic index. You can deliver a lower dose to the patient. You don't get the systemic presence of the gene therapy vector in the bloodstream, which can potentially cause side effects and immunogenicity and so on. So some kind of inhaled therapy directly to the lungs would be incredibly useful for all these diseases. And to just give a kind of case study of one particular disease, I'll talk about probably the archetypal best characterized disease for which people are targeting inhaled gene therapy, and that's cystic fibrosis. So CF is a lung disease caused by inactivating mutations in the CFTR chloride channel. So the way this works is in a healthy lung, you have a layer of mucus coating the lung epithelium, and then this layer of mucus is continuously cleared by mucociliary clearance. But the important thing about the properties of this mucus layer are that there are CFTR chloride channels in the lung epithelium, which allow for chloride efflux. Water follows by osmosis. And this allows for the mucus layer to be hydrated, which allows for mucociliary clearance. So when this is constantly cleared, if you inhale some kind of pathogen, it just gets cleared away and your lung stays relatively healthy and sterile. Now in the CF lung, there's no chloride efflux, and so there's no water efflux. And so the mucus layer gets very thick and static. Mucociliary clearance doesn't occur. You can, if you inhale a pathogen, instead of getting cleared, it divides, can form biofilms, and you get a vicious cycle of infection and inflammation and eventually lung failure. So while the disease itself has a pretty complex, has pretty complex downstream effects, it's in theory really, really simple to cure, right? If you just restore CFTR function, restore chloride efflux, you've basically cured the lung disease. Now, the problem is that while it's very simple, it's very much not easy. So the CFTR gene was discovered in 1989, and pretty quickly people decided, oh great, this is going to be really easy. Now that we know what the gene is, we just got to do some gene therapy, we're going to cure the disease, it'll be cured by the time the year 2000 rolls around. It hasn't been that simple. So we've had now 30 plus years of inhaled gene therapy efforts. So for example, people first tried to do adenoviral vectors for inhaled CFTR gene delivery. This didn't really work. Probably poor transfection, potential immunogenicity concerns. AAVs also turned out not to work when inhaled, potentially due to limited cargo size, also lack of efficacy, and again, immunogenicity concerns. Although people have restarted clinical efforts recently, and we're going to see how that goes. Other kinds of viral vectors like lentiviruses have had other kinds of safety, potential safety and efficacy issues. Non-viral delivery started with liposomal DNA delivery. But again, that hasn't really been going very far, mostly due to a lack of efficacy, potentially also due to the challenge of nuclear entry when you're delivering DNA, that DNA has to get into the nucleus. So more recently, as we're all probably aware, mRNA loaded lipid nanoparticles have kind of gone to the forefront of non-viral based gene therapy. And so there actually has been one inhaled gene therapy clinical trial that's posted results. One inhaled mRNA LNP trial, and it did show a lack of efficacy, though it did also show safety. So again, this problem of inhaled gene therapy, if we could crack it, we'd be talking about treating this huge range of diseases, CF, but also all these other diseases for which we have really clear candidates for what gene we'd like to deliver. But we're just not getting the genes into the cells. But while the first attempt with mRNA LNPs didn't necessarily work like hoped, LNPs and other organs have shown a tremendous amount of clinical and preclinical promise that makes us think that this is still an approach worth targeting. So in the liver, Onpattro is an FDA-approved siRNA LNP, and there's also been quite a bit of success with intravenously delivered CRISPR-Cas9 gene editing. And then, of course, the vast majority of us have probably gotten intramuscular vaccines based around mRNA LNPs. That's the Moderna vaccine and the Pfizer-BioNTech vaccine. And then there's all sorts of other clinical and preclinical efforts ongoing for things like targeting T cells, hematopoietic stem cells, spleen, splenic immune cells, and all kinds of other tissues, including hopefully the lung epithelium. All right, so this is kind of a problem. We really want to transfect lung cells, and this is hopefully the solution, but there's still a lot of work that really needs to be done, mostly around this problem of gene delivery potency. Up till now, people have inhaled stuff, they've inhaled genes, and the genes haven't gotten into the cells. So how do LNPs get the genes into the cells? So it's really mostly based around this secret sauce, these so-called ionizable lipids to help LNPs deliver RNA to cells. So an LNP is comprised of some kind of nucleic acid you're trying to deliver, along with various helper lipids, and the key secret sauce, which is the ionizable lipid. You mix these together to make a nanoparticle. The nanoparticle has to get taken up by cells via endocytosis, and then perform endosomal escape, and then the RNA can be translated into whatever functional protein of interest. So ionizable lipids basically are what they sound like. So here's the chemical structure of SM-102, which is the ionizable lipid for the Moderna vaccine. So what you'll see is an ionizable head group comprised of the tertiary amine and an OH group, and then a hydrophobic lipid tail. And if you, when I was really interested in taking a look at a lot of these either FDA approved or very advanced preclinical or clinical lipids that different companies are advancing. And these lipids were designed using rational design based on the principles known to help with things like endosomal escape. And what I noticed is that a lot of these basically have very, very similar themes to each other. So if you take a look at these different chemical structures, you see that tails are pretty much always either unsaturated or they're branched linear tails. And then sometimes people rationally add in OH-containing head groups. And then typically you'll add in biodegradable groups, things like esters or carbonates. And so this is true for rationally designed lipids, that they tend to be relatively straightforward variations on this theme, though you might use an ester or a carbonate or different lengths of lipid tails. And the same is actually kind of true for less rationally designed, more high throughput screening-based approaches to designing lipids. So for example, here's some work from our lab published last year, where we synthesized about 500 different ionizable lipids using a 3-component reaction between an amine, an isocyanide, and a ketone called an Ugi reaction. So you put you take basically one from this panel of amines, one selection from the panel of isocyanides, one selection from the panel of ketones, and that'll react together in a predictable fashion to make an ionizable lipid. But the thing is that you rationally design the components, and so you end up with a lot of the same common features that you'll find in the rationally designed ionizable lipids, things like branched and unsaturated tails and normal biodegradability. So the question that I wanted to answer is whether we could look at different new portions of chemical space and see if we could find nanoparticles that might be even more potent or have other kinds of interesting or useful properties. So the way I want to situate this is in this kind of Venn diagram here. If you look at the space of all possible synthetically accessible ionizable lipids, there's going to be some fraction of them that are synthesizable via high throughput chemistry, like the combinatorial reaction that I showed on the previous slide. And then a different but overlapping fraction is going to be the set of lipids that I would call rationally designable, which are ones that have the classic branched tails, unsaturated tails, linear tails, and so on. And then an overlapping subset of those is going to be the ones that are actually useful, because not all things that we think are going to be good are good. Not all things that we can make with high throughput chemistry are good. And so the two strategies that people have tried in the past are this rational design approach, where you make a few lipids in a very targeted fashion. Usually a high fraction of these turn out to be useful, but you have a relatively limited chemical space because these things have to be selected by expert chemists. And then you can do experimental screens that tend to be, again, focused more on things where the components are rationally designed. The question I wanted to answer is whether we could look at doing some AI-assisted design instead. So potentially by training a deep learning model to design ionizable lipids, we might go to different regions of chemical space that might unlock new levels of potency or other kinds of useful properties in terms of safety, or different tissues or cell types targeted. So in order to do this, I developed something that I call LION, or lipid optimization using neural networks. So the way this works, it's a deep learning model where the model architecture takes into account two features of a lipid nanoparticle, or a lipid nanoparticle, I guess you could say nucleic acid delivery measurement. So property number one is the chemical structure of the ionizable lipid. And this gets fed into something called a message passing neural network to kind of process the chemical structure into a useful vector that can be kind of learned in a deep learning model. And then also the metadata, things like the formulation parameters, that's all the other components of the lipid nanoparticle, the helper lipid identity, the ratios of the different components. So all of this gets fed into a dense feed-forward neural network and outputs a predicted delivery of the nucleic acid of interest. And so the way we use this to screen stuff is we take a trained LION model and then just list out all the possible ionizable lipids that could in principle be synthesized, have LION rank them, and then synthesize and test the ones that the lipids that LION likes the most. So this is great, but the first thing that we need to do is actually train a model, because any machine learning model is only going to be as good as the training data that gets put into it. So in order to do that, I went and I scraped the literature to find every different data set that I could that shows high throughput measurements of mRNA or siRNA or whatever nucleic acid delivery. And so here, I'm not going to be quizzing you on the structures, but what I want you to take away is that these are representative structures from different high throughput LNP screening data sets, and they're very chemically diverse from each other, which means that the input data to the model has all kinds of different chemical structures, and that's hopefully going to allow the model to learn generalizable rules of ionizable lipid design that are going to come in useful when designing my own, our own lipids that are really chemically distinct from what's been seen in the training set. So the first thing you do when you're training a deep learning model is you take a held out test set and you look at the performance on the held out test set. So that's what I'm showing here. So the way this works, this is with using 5-fold cross validation. So I train the model on 80% of the data, and then I test the performance of the model on the remaining 20% of the data, and you do that 5 times, each with each block of 20% of the data. And what I'm showing here is the correlation between predicted delivery and experimental delivery for each held out test set and for each particular data set that I scraped and trained the model on. And so a correlation of 1 means that the model is predicting delivery perfectly. Correlation of 0 means it's doing absolutely nothing. And generally speaking, the correlations are above 0. So we're getting pretty substantial ability to predict the delivery of unseen new ionizable lipids. Okay, so this is nice. This thing works in silico, works in the computer. Can it actually design new stuff? So in order to do that, we have to have an actual space of ionizable lipids to look at. So to do that, I went to my friendly neighborhood chemist, Idris Raji, and he came up with this wonderful new chemical approach that was extremely chemically flexible, and it was able to make any of around 1.6 million ionizable lipids with commercially available components. And obviously, we can't make 1.6 million lipids, but I can use LION to sort through and make the ones that LION likes the most. So we did that. After an initial round of screening with about 30 or 40 different ionizable lipids, we came up with the top hits that are shown here. And these are tested for intramuscular delivery of luciferase mRNA. So higher signal on the y-axis is better. Basically, this is sort of a mRNA vaccine type of model here. And we're comparing to the two controls, SM102 on the left, that's the Moderna mRNA vaccine, and CKK12, which is sort of a preclinical, extremely potent LNP candidate. And so what we're seeing here is a lot of these candidates are good. They're sort of right around just a little below SM102, which is FDA approved. So that's pretty impressive, but we don't want to be as good as or a little worse than SM102, we want to be better. So taking a careful look at the chemical structures, we are able to make a single rational modification to a couple of those previous lipids to end up with FO32 and FO35 in particular, and also EB66 that are outperforming the controls. And that's true across 4 orders of magnitude of different doses delivered intramuscularly. So we're just seeing across the board, FO32 and FO35 are outperforming the controls. So that's going to make these really interesting mRNA vaccine candidates. But I promised you pulmonary gene therapy, not mRNA vaccines. And so then I wanted to look at the delivery to the respiratory tract. So the next thing I did was I looked at intranasal delivery. So here you have the mice just inhale nanoparticles into their nose. These will look at luciferase expression in the nose. This is kind of what the mice look like when they're imaged. Their noses light up with luciferase expression. And FO32 and FO35 either outperform or are comparable to every control that we could throw at it in the nose. And then we wanted to look at more of the deeper lung, the more distal airways. And to do that, we looked at oropharyngeal delivery, which is like intratracheal delivery, if you're familiar with that, without a catheter. Basically, you have them inhale liquid containing LNP deep into their lungs. And then you look at the luminescence of the lungs. And here, FO35, and especially FO32, basically outperform everything we could throw at it. FO32 by somewhere between an order of magnitude and a factor of around 3. It outperformed every different control that we were able to test. Now, in people, we wouldn't be doing intratracheal delivery. We'd be doing nebulized delivery, where you administer a mist to a patient using a nebulizer. It's a very gentle process, except it's not super gentle for the LNPs. They can tend to aggregate. But by nebulized delivery, we found that FO32 was able to get comparable delivery to this IR11717, which we'd previously published as being specifically optimized for nebulized mRNA delivery. So that's very exciting. But then the question becomes, all right, that's great. We can make mouse lungs light up. What does this actually mean? Mice, are not people. So I want to back up a little bit and talk briefly about lung anatomy, because it's important for understanding the results I'm about to show you. So when you breathe in, if you look at the lung anatomy, it's sort of composed of a little bit like 2 structurally and functionally distinct or suborgans. So there's the conducting airways, and that's the place that air flows through when you breathe in. And that's the part that's mucus secreting. It does mucociliary clearance and so on. It doesn't do gas exchange, the part where blood gets oxygenated and CO2 goes away. That's the respiratory airways, or the alveoli, as you've probably heard of them. And so the interesting distinction here, the respiratory airways and conducting airways are important for different diseases. So for a disease like CF or COPD, you're really interested in the conducting airways, and mice are not really great models, but ferrets are very, very good models of the conducting airways. Meanwhile, the respiratory airways are good models of things like idiopathic pulmonary fibrosis and other kinds of sort of more deep lung diseases. Okay, so we want to see if we can transfect these large animal models of these ferrets. And so here we collaborate with John Engelhardt's group at the University of Iowa that has this wonderful Cree reporter ferret. And the way this works basically is cells express TD tomato unless you deliver Cree mRNA, at which point you get recombination, and the cells turn from red to green. So everywhere that you see a cell turn from red to green, that's a transfected cell. So the LNPs were administered via intratracheal microsprayer to the ferret lungs. And then 2 weeks later, they imaged the lungs on histology, and we see widespread green. So that's great. There are some places that are all red. This is due to patchy deposition during the microsprayer process. So places that are all red just didn't really receive any droplets containing LNP. But in places that did receive LNP, there's pretty robust transfection, which is nice. But now we want to see where specifically in the lung we're transfecting. So FO32 and FO35 both transfect alveoli. That's the kind of squiggles, the sort of tight squiggles that you're seeing are alveoli respiratory airway type structures. And so we are observing in different places that both of these LNPs can potently transfect alveoli. Now, alveolar transfection has been reported with things like polymeric nanoparticles in the literature. What people haven't reported is transfection of the conducting airways, but we did actually observe that, which is very exciting. So here we're looking at transfection of both the bronchi and the trachea, different portions of the conducting airways. Basically the bronchial and tracheal epithelia are turning from red to green, which is very exciting. That's what we want to see. we're getting about 30 to 40% of cells transfected, which is well what we would need to treat a lot of most of these different diseases. And then another of the really exciting findings that we had was that FO35, if you look on the bottom left, is turning some cells greener than in these kind of little circles or loops. So those are submucosal glands, which are incredibly important for airway biology. They secrete different kinds of mucus. They secrete antimicrobial peptides, important for airway homeostasis, and so on. So transfecting those is really important for a lot of airway diseases. And so the ability to do that is, I think, pretty unique with these nanoparticles with the testing that they've done. So just to conclude, at the beginning, I told you that I hoped that we are going to get LNPs that were both useful and potentially outside the beaten path for ionizable lipid design. And unfortunately, I can't show you the structures quite yet. So stay tuned for when this thing comes out. But so you'll have to take my word for it for the next month or so that FO35 and EB66 do in fact have these very weird structures that one wouldn't necessarily think about synthesizing. So the fact that we were able to get especially FO35 to get the state-of-the-art delivery and the submucosal gland transfection in a large animal suggests that there's really chemical spaces that haven't been looked at in the past, but really can do really interesting things therapeutically. And so to give the final conclusion, we designed these lipids that beat or match state-of-the-art. Basically in every local delivery context, we're able to test in the mouse. And then we're also able to scale up to these large animal ferrets. And hopefully we can get some more large animal data soon. And with that, I'd like to thank my collaborators, Rajiv, Idris, Alan, Sandra, Emily, and Elise, as well as Dan and Bob, and Tara. And thank you.